Hi dear children, today in this topic we are going to study in detail about photosynthesis. That is a complete process of photosynthesis. Already many things we have told in the introduction part itself. So altogether we will be explaining that. So listen it carefully. Okay, so let us start. See, we have already studied the structure of chloroplast, right? From the thylakoid, that is the individual unit of grana, from the thylakoid, actually, the photosynthetic action begins. We have studied light reaction as the first step in photosynthesis. So it starts from the thylakoid of the chloroplast. Each thylakoid is a membranous structure, and inside the thylakoid there is a cavity space called as lumina thylakoid. Now, when we look into the inside the thylakoid contain all the pigments. All pigments are seen inside the thylakoid, right? And these pigments can be seen in the membranes of the thylakoid and also inside the lumen of the thylakoid. So it is disposed inside the thylakoid. Now, all the pigments in the thylakoid together they form a complex structure with the protein. It's called as light harvesting complex. That is, all the pigments in the thylakoid together they form a complex structure which is called as LHC. Okay. Why it is called as LHC? From the word itself you will understand the meaning. It's light harvesting complex because these pigments in the thylakoid trap the sunlight and carry out the light reaction in the photosynthesis. That is why it is called. Now, in this light harvesting complex, there are certain pigments which have the ability to accept the or to trap the sunlight with a wavelength of 700 nanometer. Such groups of pigments in this light harvesting complex, they are specially called as a system called photosystem 1. Same way, there are another group of pigments in this light harvesting complex that is inside the thylakoid, which can trap the sunlight with a wavelength nearly the wavelength of 680 nanometer. So such pigments together they also form another system which is called as photosystem 2. These are nothing but they are the subdivisions of the light harvesting complex. Light harvesting complex itself is the whole pigment in the thylakoid which forms the complex. In that one group of pigments they can trap sunlight with one wavelength and another group of pigments can trap the sunlight with another wavelength of light. On that basis we have divided light harvesting system into two groups like photosystem 1 and photosystem 2. This 1 and 2 is denoted not based upon who is functioning first, it is based upon their discovery. Photosystem 1 was discovered first and photosystem 2 was discovered later. Now, actually this group of pigments forming photosystem 1 and 2, we can see towards their membranes of thylakoid. Okay, not towards the center, they are seen towards the membrane of thylakoid. Now let us see what is the speciality of these two pigment systems. In two pigment systems, one, because we know that they are containing the different type of pigments like chlorophyll A, B, carotenoid, C, D and so on. In this, one of the chlorophyll A molecule of these two systems, രണ്ട് സിസ്റ്റത്തിലെയും ഒരു chlorophyll A molecule, കുറേ chlorophyll Aയും Bയും Cയും ഒക്കെ ഉണ്ടാവും. One chlorophyll A molecule of both systems will act as a reaction center. That is, one of the chlorophyll A molecule of each system will start up the reaction. Light reaction will be started by this one chlorophyll A molecule of each photosystem. Photosystem 2 ആണ് ഫംഗ്ഷൻ ആവുന്നതെങ്കിൽ അതിലത്തെ ഒരു chlorophyll A molecule ആയിരിക്കും ആദ്യം ആക്റ്റീവ് ആവുക. അവരാണ് സോളാർ എനർജീനെ ട്രാപ്പ് ചെയ്തിട്ട് അവിടെ റിയാക്ഷൻ സ്റ്റാർട്ട് ചെയ്യുന്നത്. ബാക്കി പിഗ്മെൻറ്റ്സ് ഒക്കെ അതിൻ്റെ കൂടെ നിൽക്കും. സെയിം വേ ദിസ് ഹാപ്പൺ ഇൻ ഫോട്ടോസിസ്റ്റം 1 ഓൾസോ. ലെറ്റ് മീ ടെൽ വൺ മോർ ടൈം. തൈലക്കോയിഡ് എന്ന് പറഞ്ഞാൽ ഇൻഡിവിജ്വൽ യൂണിറ്റ് ഓഫ് ഗ്രാനയാണ്. അതിൽ നിറയെ പിഗ്മെൻറ്റ്സ് ഉണ്ടാവും, പല പിഗ്മെൻറ്റ്സും ഉണ്ടാവും പ്ലാന്റ്സിൽ. ഈ പിഗ്മെൻറ്റ്സ് എല്ലാവരും കൂടി ചേർന്നിട്ട് ഒരു കോംപ്ലക്സ് ഫോം ചെയ്യുന്നുണ്ട് വിത്ത് ദ പ്രോട്ടീൻ. ആ കോംപ്ലക്സിനെയാണ് നമ്മൾ LHC എന്ന് പറയുക. അതായത് ഒരു ഗ്രൂപ്പ് ഓഫ് പിഗ്മെൻറ്റിനെ തന്നെയാണ് നമ്മൾ LHC എന്ന് പറയുന്നത്. അത് നമുക്ക് അഗെയിൻ സബ് ഡിവൈഡ് ചെയ്ത് 2 ഗ്രൂപ്പാക്കി തിരിക്കാം ബേസ്ഡ് അപ്പോൺ ദ ഇൻറ്റൻസിറ്റി ഓഫ് വേവ് ലെങ്ത് ദാറ്റ് ദേ ക്യാൻ ട്രാക്ക്. അതിൽ 1 ഗ്രൂപ്പ് ഓഫ് പിഗ്മെൻറ്റ്സ് വിച്ച് ക്യാൻ ട്രാക്ക് 680 നാനോമീറ്റർ വേവ് ലെങ്ത് സൺലൈറ്റ് ഉള്ളതിനെ നമ്മൾ ഫോട്ടോസിസ്റ്റം 2 എന്നും, അനദർ ഗ്രൂപ്പ് ഓഫ് പിഗ്മെൻറ്റ് വിച്ച് ക്യാൻ ട്രാക്ക് സൺലൈറ്റ് വിത്ത് എ വേവ് ലെങ്ത് ഓഫ് 700 നാനോമീറ്ററിനെ നമ്മൾ PS1 അല്ലെങ്കിൽ ഫോട്ടോസിസ്റ്റം 1 എന്നും പറയുന്നു. ആൻഡ് ഇതിൽ ഓരോ സിസ്റ്റത്തിലും ഒരു ക്ലോറോഫിൽ A മോളിക്യൂളാണ് അവിടെ റിയാക്ഷൻ സെൻറ്ററായിട്ട് ആക്ട് ചെയ്യുന്നത്, മീനിങ് ദേ വിൽ സ്റ്റാർട്ട് അപ്പ് ദ റിയാക്ഷൻ. Here one more thing is there. This LHC or the light harvesting complex is also called as antennae of the chloroplast. You know what is antennae? Antenna of TV and so on. You know what is the function. Same way, the light harvesting complex is also called as antennae of the chloroplast because it starts up the light reaction. Let us start to say how is this chemical reaction, that is the light reaction happening in the chloroplast. We know there are 2 systems like photosystem 1 and 2, but here the photosynthetic process, that is the light reaction, starts with photosystem 2. Then only photosystem 1 will become activated. So here the first role is done by photosystem 2. The reaction center in the photosystem 2 will, what they will do first, they will trap the sunlight with a wavelength of 680 nanometer, and the electrons which is seen in this photosystem 2 will get activated. As a result, they get excited and they jump out. These electrons will be accepted by an electron acceptor. Here the electron acceptors are the cytochrome proteins. They accept the electron. Now this electron from the first electron acceptor get transferred to the next electron acceptors. Likewise, there is a formation of a chain of electron acceptors to carry the electron to the next photosystem, that is photosystem 1. So we can see an electron transport chain with cytochrome forming a system, and thereby electron reaches to photosystem 1. Now you can see one thing here. When the electrons was transferred from photosystem 2 to photosystem 1, now the photosystem 2 is vacant with electron. Since this is a continuous process, that should not happen. Photosystem 2 which starts the reaction should not remain free without electron. So there, how electron will be made available? So once that electron is moving from photosystem 2 to 1, again the electron should be there in photosystem 2, and one by one after the other, the electron should continuously move. Then only the photosynthesis process will be carried out successfully. So how? And again electron will be made available to photosystem 2 to continue this light reaction. The role of water molecules. The water molecules which will be seen in the lumen of thylakoid at the time get splitted out with the help of solar energy. With the help of sunlight, this water molecules split and they form hydrogen and oxygen, and electron is released. This electron is being transferred to the photosystem 2, and in this way always in photosystem 2 there will be presence of electron for carry out the light reaction. Let us see what will happen simultaneously at this time in photosystem 1. We have already told the electron has been excited and moved to the photosystem 1 from photosystem 2. And now what happens in this photosystem 1? Now the same process happened in photosystem 1 also. One of the chlorophyll A molecule which forms the reaction center get activated by the sunlight with a wavelength of 700 nanometer, and they make excited this electron which is seen in the photosystem 1, and this electron, once it get excited, it also jump out and they will also get accepted by the electron acceptors. Here also the same, like there are chains of electron acceptors. They will accept the electron and the electron move one by one, and finally they reaches an electron acceptor which contain an energy unit called NADP. They accept the electron and this NADP reduces to form NADPH. Here, children, this is the whole light reaction process which happens during photosynthesis between photosystem 2 and 1. Now this whole process is actually called as non-cyclic photophosphorylation, in the sense like the electron which is transferring from photosystem 2 to 1, it's never returning back to photosystem 2 itself. That is why it is called, the whole process of light reaction called as non-cyclic photophosphorylation. Photophosphorylation in the sense ATP production is there when the electrons are moving from one system to 2. That we will explain in another topic like chemiosmotic theory. Then this process is also called as Z scheme of light reaction, in the sense there is an Z shape in the movement of electron. You can see there is an Z shape of movement of electron. Okay children, I hope it is very clear to you now. Okay, thank you for watching my video. Okay, if you like my video, as we always say, please subscribe and support my channel, and also press the bell button so you can get easy notification of the next classes. Okay, thank you once again. Have a nice day. May God bless you all. Thank you.